Good morning, Thomas.
Hi, very good morning.
We do have quite a few people who have already joined, so we may as well begin on time for once. What do you think?
Yeah, absolutely fine for me. It's already
Okay,
late in
great.
the afternoon.
Right. Right. Yeah. So okay, then I'll get started with your intro, which is like 1 page long. I started writing it just before this webinar today. And for the viewers who have joined this webinar, uh, Thomas Altman is he has more than 20 years of experience in terms of cleaning validation. He he works as a global technical consultant with Ecolab. There is not a single guideline in cleaning validation which he has not contributed to. Uh, in terms of PDA. I think currently, Thomas, you're working on ASTM standards.
Yeah, correct.
Um, yeah, and I'll probably not go into all possible details, but thank you so much for joining this webinar today, and welcome.
Thank you very much, Vivek. Thank you for the nice introduction.
Yeah, and just before we begin the webinar, the topic of today's discussion is all about FDA compliance. Thomas has kindly agreed to talk about the top issues which we continue to see with the US FDA inspections, and we have analyzed quite a bit of data. We'll go into that in a moment. But today's session will be all about what are the top pain points and problems that the pharma industry continues to see even today in terms of cleaning validation. We'll look at the top 5 of them. But what I think would be really valuable, which Thomas would do, is talk about how to actually solve those problems, and before that, why do some of these problems even happen in the first place. So I am personally very excited, and Thomas, we do have a lot of people joined now, so let's get going. I'll share my screen.
Yeah.
Just
No, and thank you for setting up that time. I think it's really good to sometimes review a kind of what is hot topics at the moment. And your suggestion for this kind of short talk, I think is really valuable, I hope, for a lot of people in the audience.
right. So what we are looking at right now is the data from FDA observations. And we have gone ahead and analyzed actually more than 7,000 observations since 2018. And we did apply some methodology to figure out what the top 5 FDA problems are. So while this data goes back all the way till 2018, we also did like a more recent analysis, and the ranking does not change a lot, which is a good news as well as a bad news. Um, so this would form the foundation for our discussion today. I'll just
Yeah,
quickly take a minute to summarize what these observations are. So there is a widespread uh, observation around not having adequate level of cleaning validation done in the facility, which means that some equipment got missed out, some products got missed out. So not having enough coverage, some criteria got missed out. So just and typically we saw that it's happening with generics pharma company a little bit more, where the number of molecules are just a lot higher. So that's one definitive pattern which we continue to see. Second point, kind of correlates to that, where uh, whenever a facility has a large number of molecules, you can't possibly go and do validate all of them. So, having an application of the worst case methodology is, something that FDA now expects in terms of risk assessment framework. So there are some problems with respect to that, which actually continues nicely to the third point, which is missing out on a proper risk assessment of hardest to clean sampling locations. And then we go into the change assessment impact in terms of one of the common issues was the campaign frequencies not being validated, and as well as releasing equipment without properly checking if all the basis is covered or not. So while these are the top observations in terms of US FDA inspection, there are many more. Uh, but I do want to spend about a minute just to talk about how did we gather all of this data. So on my screen, I have FDA tracker.ai, which is a software tool which helps us analyze the FDA observation in a little bit easier manner. And we'll just head over to the FDA 6 systems. And we see that cleaning validation has been amongst the top 5 for a while now. As of today, it stands at number 3. And if we just head over to the types of observations which are typically found, I mean, there is just a lot of them which we have gone through in an algorithmic manner. And just to take an example, an observation was given in terms of equipment and utensils are not cleaned at appropriate interval to prevent contamination. So this is a standard phrasing which FDA would typically use. However, we can go ahead and very quickly figure out the exact problem as well. So in this case, there was an inadequate cleaning validation coverage, particularly in hard to clean areas, which actually goes back to our point number 1 and point number 4 in the top 5 US FDA observations. We could analyze this issue even further in terms of what are the potential root causes, what are the typical corrective and preventive actions, and what compliance checklists we can follow to ensure that this issue does not happen in the future. So we used this particular tool to analyze a vast amount of FDA data, and I would actually be towards the end of this webinar, which would go on for approximately half an hour. We wouldn't want all of you to stay beyond that. I know many of you will probably have meetings booked after half an hour or so. So we'll try to wrap this up. But before we finish, I'll just very quickly talk about how you can access this tool for yourself for free. So anybody who has joined this webinar, we would be giving away this tool for free. So just wait for another 20-25 minutes or so, and we'll tell you how you can yourself sign up for this tool as well. So with that, I'll go back to
And I
our.
think, Vivek, that was a perfect example, because for me, this kind of data, what is digitally available, to use it in tools in AI is the future, for even for cleaning validation. Because this is what we see here also on the FDA observations. They publish it and you can really evaluate it. I'm sometimes asked, oh, how about Europe? You're located in Europe. What about the findings there? They are not published by the regulators. So this is why it's so good if you have these data to analyze it and to use modern technology to do it. And this is also what the pharma industry should do in the future. If you get data, use it.
Absolutely, and rightly said about the EU regulations. Even though FDA is just a lot more flexible, transparent. It publishes data more easily. And there is some data available about EU, but there is a bit of a lag, and sometimes it's just not available in as much detail as we would, which would become helpful to the industry. So hopefully we see a little bit more improvement coming up with that. But let's just I just wanted to quickly summarize these 5 problems again before we start heading over to the solution. And Thomas, when we talk about these problems one by one, one other thing uh, which we would talk about is how can some of those problems be mitigated by using software solutions, digital enablement, and just a little bit more automation, which can take away the pain. So this is essentially, all the things that a pharma facility will need to do in order to set up like an end-to-end cleaning validation lifecycle. Many things can go wrong in following this, but today we'll touch upon the top 5. So all right. So coming to the first one, which is all about, in my opinion, setting the right foundation for the cleaning validation in terms of master data, creating your product equipment matrices, setting up the cleaning validation policies, worst case ranking, risk assessment principles, and a ton more things which are typically found in any cleaning validation SOP. uh, you know, and those are designed based on US FDA regulations, EMA guidelines, which has been advancing at a little bit faster rate than US FDA, as well as some of the pioneer work which Thomas, you and some of your colleagues are doing at ASTM and PDA. So, with that, Thomas, would you want to talk about this observation in a little bit more detail?
Yeah, cleaning procedures not validated. Sometimes people ask even, how can this happen? So how is it possible that in pharma something is not validated? And there can be some reasons for that. And what I've seen lately is, for example, that people are having multiple equipments and they just missed out to validate a certain cleaning for a new equipment, or they have put it on hold as this is not a priority at the moment. And in the next inspection, they are even not aware of, oh yeah, that was outstanding. We did not really complete this. And this is something where I would always say, if you are having a cleaning procedure, you need to ask yourself, how is the validation done? And did I do it already? And if I have it in place, how can I maintain that the validation status stays? And is there a verification required? Or these kind of questions is what I would say very much important. But the thing that, oh, the cleaning procedure is not validated can really happen. And this is just due to the fact that you have so many different equipments on site and you have priorities. You want to validate first your main equipments with what provides the best performance or even the highest throughput. And then you are going to say, after that prioritization, I cannot do everything at the same time. And this leads then to the fact that at the end of the day, there might be a cleaning process not validated when there is an inspection upcoming.
Yeah, and Thomas, just to add some more data to this observation, uh, the cleaning procedures not being validated, boils down to multiple root causes. But we saw that there are predominantly 2 of them. The first one is deficiencies in design, and the second, can be at a high level classified as deficiencies in execution. So there are roughly half of the cases where the SOPs were not written uh, adequately, because US FDA regulations have been, or at least the expectations have been evolving rapidly. I think especially after the EU regulators moved to health-based exposure limit, and some of the industry bodies like ISP, they published another guideline which mimics that uh, uh, expectations. We know that PICS followed immediately afterwards. And now with the ASTM work as well, which is considered to be very close to what FDA would sometimes think.
Yeah.
Uh, just the expectations of the regulators have been evolving so rapidly that there has become a gap between what's expected and what's in the SOP. So that's deficiency number one. But the other deficiency is that even though you had the right policies in the SOP, but just the execution could not happen properly. For example, uh, as simple as the hardest to clean sampling locations. So while the SOP might say that you need to apply like a quantitative risk-based criteria, but that just did not happen. So there is a definitely more flavor to this problem. But by and large, it can be attributed to, I think some problems at the design level and then some problems at the execution level.
Absolutely,
But
absolutely.
yeah, but Thomas, thank you so much for, sharing that
Mhm.
examples which you did, which I think, then I think you did already talk about the worst case bracketing a bit, but in your opinion, what are some of the problems that, because knowing you, I know that I always find you at some airport, you're always visiting customers, you're always speaking with people who are doing, solving these problems day in and day out. So it would, I think, be a lot more beneficial if you share your experiences of what you keep on seeing in the industry in terms of the worst case bracketing problem.
Yeah. Yeah, so here I would really say that this is a kind of activity what we are doing since the last 2 years a lot, because we've seen that a lot of people say, oh, my cleaning processes are validated for a long period of time, but we never reviewed it really. And we need to go now into the, I would say, outline that the cleaning must be verified. We need to understand the cleaning, and therefore we need also to understand what is our worst case product. Because in the introduction, you already said, you do not want to validate all your products. And it's even legally outlined that you can identify the worst case and then do the validation for your worst case product. But then you look into the data you have. And what I can see in the market is that people who are dealing with cleaning validation are often new to the company or just coming in from a university or something like that. And their first projects in the pharmaceutical industry is then, oh, you get a cleaning validation project. And then to understand what people have done, maybe even 10 years ago. I've been to a site where they said, okay, we have validated this process 10 years ago, and now we want to revalidate some things. And they needed to understand, oh, what has been done 10 years ago? And the cleaning itself, why is it set it up as it is? And then we were talking, how was the consideration on your worst case product done? And they were looking and said, okay, we have looked at solubility and toxicity, and that's it. I said, yeah, but that's not really true these days anymore. If you look to the guidance documents, you should involve cleanability. And here I would strongly suggest talk to your operators who are carrying out the cleaning to identify, oh, where do I have troubles with cleaning? Then you get a good outline of the risk assessment or for the risk assessment where you can maybe have difficult to clean products. It's not enough just to look at the solubility and maybe the toxicity. Yeah? There are a lot of creative ideas at the moment to make a consideration on worst case products, and that was what the regulators are expecting. You have made your mind on different aspects for all your products on site, and you really want to make sure that you are validating only the worst case.
Yeah, and unfortunately, there is no one single method which works for each facility. It really depends on what you are manufacturing. If you are doing
Absolutely.
high potency APIs, you'll end up doing something different versus if you're manufacturing topicals. And this is exactly the reason why we had to build, I think, more than 20 plus risk parameters. Thomas, you would remember when we had a discussion around that. So every
Yeah.
company will need to perform that risk assessment, figure out what those criteria are, which affect severity of harm, the probability of occurrence of harm, and as well as the detectability, which is the standard risk framework which US FDA recommends.
Exactly. And you need to document that. You need to verify in a system, like in a digital system or wherever, how you are making this kind of decision. Now, I have customers who are discussing, oh, if I produce a product very often, there is a high risk that I have a lot of patients getting a defect by a product when something goes wrong. Other products, our other customers telling us, if I produce a product a high amount of times, I have really a routine in production. So I don't see this as critical as a product what is very seldom produced because there is no routine. So these kind of thinkings needs to go into the documentation and needs to be really documented safely, that in case of an inspection, you can show your considerations. And this is not only today, also in 3 years from now, when you get an inspection, you need to have this information on hand.
And I wish we could continue talking about this topic, Thomas, a bit more, because there is a lot more to be discussed here, especially in terms of we haven't touched upon the coverage. We haven't really talked about the risk ranking. So this, I think this topic itself truly forms the foundation specifically for large volume pharma manufacturing organizations. But uh, you know uh, moving on to the third top issue, which actually was number one issue at some point. I think about when we had looked at this data perhaps 18 months ago. This was indeed the number one problem at the time. I think ever since ISP did some work in this area, companies have now picked it up and is gradually becoming slightly more easier to follow. But there are so many examples I've heard from you, Thomas, around uh, the companies really struggling, especially with the documentation around managing hard to clean sampling locations.
Exactly like that. And if you ask people often for the how did you identify your hard to clean location? They say, oh, I passed with this equipment a riboflavin test, so everything is good. So they do not really have the identification done in a way that they might think of, oh, where is the drying on residue the most because there is maybe a heat exchanger, or where is the idea for me to have the worst case for cleaning because in the top then it's drying on much longer than in the bottom of the tank or something like that. And at the end of the day, we know that for validation, a lot of samples were taken. And I can tell you that I've been on sites where they say, oh, yeah, now I have to sample here and there. And I was asking, yeah, is this your identified hardest to clean location? Yeah, I think so. That is somewhere in the documents, but yeah, it was always tested here. And these kind of things is to be avoided in the future. You need to have really a waterproof system that tells you, oh, this is the hard to clean location. You have maybe a picture or whatever to it, or just a schematic of the equipment, and then even the reason why this is a hard to clean location for you and why you consider this maybe for the validation purposes, so that you need to sample there if the active is staying there, or if you maybe are considering, oh, there might be leftovers on cleaning agents because this is a hard to rinse end of the line or whatever. That needs to be really documented in a way that you can afterwards show I have made considerations, and then it's even for long period of time valid, because it should be involved also with the change controls you are doing for the equipment. So, and if you don't do any critical changes, you have it really on hand for the inspections.
Yeah, and never miss the documentation, I think, is a key takeaway as well. Just one other point I would add here is so we are seeing an increase in FDA inspectors, uh, asking for a more objective, a more quantitative risk assessment now. So, while there can be a subjective justification, just figuring out what those risk parameters are. For example, if this is like a moving part, then there can always be a hot spot for contamination. And I
Yeah.
mean, we all know that the cross-contamination is a localized phenomenon, even though the samples that we take in terms of testing the critical quality attributes at the end, it might be all good, or even the swabbing that we do at the uh, at each batch or product changeover, might not have any residue. But if those localized contamination hotspots are not considered, FDA does not find comfort in the fact that there could have still been some localized cross contamination which could have gone undetected. So uh, something that we came across actually multiple FDA 483s.
Yeah. Yeah, and that's important to understand your equipment, to know where
Yeah.
residues dry on, where residues are maybe even changing due to different temperatures or due to the fact that some humidity is touching there. When you introduce into a mixer or something like that at the top, you might have then clumping of products. All these kind of things is what they want to see, that you have considered that kind of situations.
Correct. Uh, Moving on to the top observation number 4, and this could actually happen due to many reasons. Releasing equipment without proper verifications, but missing out on cleaning verification still remains one of them. And we know that in Europe specifically, we have a concept of QP, qualified personnel, and
Mhm.
it happens to be their responsibility to ensure that the equipment has gone through all the adequate cleaning verifications before the product which was made on that equipment could actually be released into the market. So we have often heard this problem that uh, it is sometimes quite difficult to get all of this data easily. You might have to go through all the historical documentation. So how do you even make sure that an equipment which could be manufacturing a few hundred products in a given 6 months period would have gone through all possible cleaning verifications that need to be in place. So it is, in my opinion, sort of inevitable that this mistake would happen. It is only a matter of when, not if, unless this decision is actually made using a centralized data inventory.
Yeah.
And we have
And you made
discussed
it
about
very
this in the past. Yeah.
No, and you made a very good point about
Yeah.
that. If you have a cleaning, you need to verify that it's carried out always in the same way. In the past, people were thinking, oh, I do it always in the same way, and I document it just with a signature that everything is fine. Today, inspectors want to see, oh, yeah, did you reach the temperature? What is the critical factor for the cleaning? They even say you need to identify what kind of cleaning process is related to what kind of cleaning attributes so that you know either it's the time, it's the temperature, it's the chemical concentration, whatever drives your cleaning. And you should verify that this is in place for the cleaning. It's not longer enough to say, oh, yeah, the cleaning has run for half an hour, you get a signature, and then the qualified person will say, oh, everything is fine. No, that should not be true for the future. And for a risk assessment, you need to identify how important or how high the risk is. Maybe it's okay to just make a visual inspection and say, oh, after cleaning, everything is visual clean. But if you have a more risky formulation or product, then you might want to demonstrate with data you get from maybe washing machines already today. Now, how long was the cycle time? What was the temperature in the washer and so on? You just need to use the data as you just described at the beginning. Yeah, the FDA shows the data and you need to use it in a way that you say, okay, I can demonstrate what are the top 5 findings. And here you would say, I can demonstrate that the washer is running as it should run. It is verified every time.
For people who might have to drop off, Thomas, I'll just jump a couple of slides,
Mhm.
and we'll talk about the free access to FDA tracker. So you can
Mhm.
actually do this today. It is valid only today. So if you go on to fdatracker.ai, you'll be able to sign up and gain access to FD Tracker for completely free. But make sure that you do this today. And there is, one more takeaway, because this is takeaway 2. So, just wait for 5 more minutes, and then we'll share, another really helpful resource, which, we receive a lot of good feedback around that. So just coming back to the, top observation number 5, which is managing changes and routine monitoring. So in any given production environment, we know that things are always moving and changing. New products, new APIs, new equipments, products getting archived, uh, some data related mistakes coming to the surface, new methods. So there are a whole bunch of changes which do keep on happening on the shop floor, and ultimately many of them have some impact on the cleaning validation status of the facility. So if not validated or assessed properly, it can lead to a compromise in the validation status of the facility. So this is all about the top observation number 5. So Thomas, where have you seen this problem around?
Yeah, this is a classical phenomenon, for example, in oral solid dosage manufacturing, where they are producing a lot of maybe the same tablets in a, just let's give it as a coating process. Yeah, they use a lot of times the same, and then it is outlined. Oh yeah, we might use it for 2 or 3 times, for 4 times, or for 5 times, and really then the outline, what is the maximum run before I need to do the cleaning. This is something what you need to identify, and you need to stick very close to it, because otherwise, if you are making a product maybe even 2, 3 batches more, that is not possible to be then cleaned effectively. So for me, this is always a kind of outline that we say, if you are looking into the cleaning frequencies or into the frequencies of cleaning, you need to also establish what is the campaign length, and this is what you need to validate. And the reason is, you are doing something today, and this situation you have might really change. Your product, what is a low mover at the moment, can be your blockbuster product in the future. So then you need to think of, oh, maybe I want to make more batches, and then is the cleaning still valid, or do I need to redo it? Or how can I make a risk assessment that it is still fine? This is the kind of verification you need to make to not come into this issue.
Yeah. And while we have discussed some of the top observations found in cleaning validation, we couldn't have possibly done justice to what all the problems are in today's session. So for the audience, what we have done, we have actually put together something called an audit checklist, which has been designed based on all the cleaning validation related observations since 2018. And you can use this checklist to perform a self audit. You can always, reach out to us for any questions. We don't do this, we don't provide this service, but we are always happy to help. But you can do a self audit using that checklist, and you can reach out to us if you have any questions, if you're finding any critical gaps. So I'll just now show how can you download this audit checklist for yourself. So all you have to do is, and it looks something like this. It's a pretty standard document that we have put together. You know it has all possible things which can actually go wrong. I mean, simple things like, do your validations include detection limits for microbial contamination? And and we know that there have been lots of problems around LOD and LOQ levels, especially when the safe carryover limits, they fall to a level that they are not even detectable by the existing method, and then you have to go into a whole new method development cycle. But we have seen in the past that by transitioning towards a more HBEL based approach, some of those reworks can be avoided. So, but in any case, we wouldn't go deeper into what all of these problems are. So, please feel free to just visit leucine.io, and under resources, you'll find an audit readiness hub, and then at cleaning validation insights, uh, just navigate to the section audit readiness checklist, and you'll be able to simply just it'll be emailed to you. Just add your first name, last name, and your email ID, and you'll be able to download this checklist for yourself straight away.
And it's a good reminder sometimes. It's just like to get into, yeah, I'm doing this since years, but do I really have considered everything? It's just to tick the box. This is why I like it, just to say, okay, is this something good for me now, or have I already thought about this process, or do we need to do something on it? I like this checklist, Vivek.
And unfortunately, it does lead to gaps. So, better maybe more proactive than reactive. Right. So I do just want to take another minute or so. So, we have seen that implementing all of these best practices that Thomas has talked about today, and with the use of, more automation-driven approach, which we obviously do at Leucine in terms of the cleaning validation software clean. We do see that there is a big impact in terms of reduction in the cleaning validation failure, things like introducing new drugs faster, ensuring that your protocols are up to date. But I do want to spend a minute on this case study. So we did see that one of our customer sites had 5 cleaning validation failures in the prior year before implementation of clean. And they were doing more of a legacy limit approach, where their limit was coming out to be 0.3 across their entire equipment train. And they hadn't really done the actual risk assessment which clean would do, based on the permutations and combinations. And clean gave limits which is in yellow, and they happen to have residue values on 5 occasions which were above their own established limits, but were actually truly below what the limit should have been. So it could have been completely preventable. We know that with each cleaning validation failure comes 200 pages long investigations, plus FDA really doesn't like to see problems with contamination with all the recent stuff which has happened. So completely preventable just by simply moving to more actual risk assessment approaches via clean. Um, and Thomas, I know, you have previously talked to me about, the similar problems we have seen in your experience as well.
Yeah. No, and I think these historical cleaning limits are often seen as, yeah, I need to reach them because this kind of idea was for me to get the worst case limit calculated. But if you then look into details, it is often the worst case calculated. So they are taking all the equipments in a process train, even if it's not used at the moment. And if you have more surface area included in the limit calculation, you lower the concentration. It is often so that you can even outline that it's not only the historical cleaning limits what is too low because you used only a safety factor, it's even also the understanding of the full process. So your process train with all the surfaces is calculated in a digital way much better compared to what you have maybe had on an Excel spreadsheet. And it's just like 2-3 weeks ago when I was on site at a customer, I was asking him, how are you doing this limit calculation? And they were telling me, yeah, we need to make this across all our equipments. And I said, but yeah, you are varying here. Sometimes you have a big batch, then you have a small batch. Yeah, we always take the worst case of the worst case. And if we are below that concentration, we are fine. And in this case, I would say they are making their lives harder only due to the fact that they are not able or capable at the moment to use other things than an Excel spreadsheet to make this evaluation, and then to say, okay, if this is the lowest number, I will stick to that number. And so this is why it's, I think, required to really think of the fact that I need some solid master data. What are my systems I'm using? What kind of limits are valid from the health-based exposure limit scenarios? And then relate this to the maximum safe carryovers. Very often you should do a comparison also, where have you been before? And then you should include the discussion. What was my worst case assumption in the past? Was it really the all over surface? Was it even all over the surface, but plus additional equipments and so on? So to get an extra safety, these kind of situations is what we see. And I think what you show here, Vivek, is just more reality data or real data, what are coming from the digital system and not longer from an assumption of a worst case.
Yeah, I mean, thank you, Thomas, for sharing that story. I know we are talking about compliance today, but the craziest thing that I saw at one of our, CDMO customer sites was that, they actually had, just one common limit for the entire plant.
Mhm.
And, the crazy part was that they were not able to introduce or try out a new product from one of their, potential customers because it just wouldn't meet the limit criteria. So it actually had a big business impact on their ability to be agile, introduce new molecules into the facility. So, if there are people in the audience at CDMO facilities, you don't need to be the worst of worst, just need to be the worst.
Yes,
Uh,
exactly. That's a good message.
so with that, I'll just summarize in terms of the takeaways for today. Obviously, we'll send out the recording. You'll have the slides, uh, and you can get access to this tool for free just for today by going on to fdtracker.ai. And as well as you'll be able to download this audit checklist. You just need to visit leucine.io. And if at all you want to learn about the cleaning validation software, Clean, which I would definitely recommend you do. So just go to this product page. It does talk about various features that we saw today, but obviously there is a lot more that needs to be done to keep in compliance with the evolving FDA expectations for cleaning validation. And if you want to learn more, just feel free to reach out to us, or there's a contact option on the website as well. So with that, we'll conclude this webinar. Thomas, thank you so much again for
Thank you.
all the practical tips and insights that you bring, along with all the regulatory experience, which I know is a lot. My last question would be, so when can we expect the new PDA technical report 29 to drop in? It's been a
Yeah.
while since we have been waiting for that.
Yes, yeah. I think we have discussed this lately in our working group, and it should be by the end of this year. So we were counting now for quarter 4 of this year.
Okay,
I
great.
hope that it will be published soon, because that goes then really hand in hand with the all the discussions we just had, ongoing verification processes, risk assessments, and also identification of sampling points and these kind of things. So
Yeah. So we do have some questions from the audience, but are there any other questions which anybody wants to directly ask Thomas? We can actually unmute you if you raise your hand, and you know I think it's a fantastic opportunity to just get any knowledge out of Thomas while you can, while we still have him on the line. So we'll just give it a minute, and I think in the meantime we do have a question around biopharma products. So there has actually been quite a few recent things on biopharma related cleaning validation. But Thomas, are there any particular differences that you would like to convey to the audience in terms of how should they manage cleaning validation when it comes to particularly biopharma products?
Yeah, in biopharma production, I think we have discussions ongoing how to really outline that the active is not longer active. Yeah, in biopharma production, you have maybe things like proteins, what are used for medical purposes, and then to identify that this is inactivated and so on. So this is something where you really need some solid data. And yeah, it is today most of the time not enough anymore just to say, oh, I have immersed my protein suspension for 15 minutes in 1 molar caustic. You need to really get into the data. What happens to the protein? Where does it break down? Or how do you can consider that this is now inactivated? These are the things in biopharma production what are more and more coming up and what I see at the moment as questions.
Great. I have my good friend, actually just before that, Dhananjay has raised his hand. I'll just see if I can uh, get him in. Yeah, so Dhananjay, you can ask your question now. And I do also see Saket, my good friend, on the call. So Saket, if you also stay there, we'd also like to hear from you for a minute. Yes, Dhananjay.
Yes, good evening, sir. I am Dhananjay from India.
Hi.
I work in a API facility. Sir, my question is regarding hard to clean area. So, for example, for a reaction vessel, let's say reactor, we are considering a hard to clean area is above the manhole of the area is considered as hard to clean. So, would you add, sir?
Yeah. I could not get the
Yeah,
last
I think
bit.
we lost him.
Okay. No, okay. Now, but this is even recorded, yeah? The manhole is classically one of the hard to clean areas, as this is something where you often introduce materials or even where you have a specific construction that if you have splashes going up to the top, they are not really cleaned with an extra spray ball or something like that. And even in API production, we still see a lot of solvent cleaning, so boiling out and so on, so that you need to consider where is my worst case spot. But I know that a lot of the people consider manholes as worst case areas because of the specific different construction compared to the rest of the equipment. And if there are some spots or splits or spillages on these areas, that they are then really not drying on over a long period of time and therefore can give really risk of cleaning failures. But I hope that this is something what he can use, as I not got the rest of it. Sorry for that.
Yeah. No, I think it definitely helps. So I'll also ask. So Saket, I know you keep on posting on LinkedIn quite a bit, and I know that you keep on speaking with many companies in this particular regard. So other than what we have discussed today, do you see any other emerging problems with respect to cleaning validation that the audience today should be mindful of?
Yes. Am I audible, Vivek?
You are, yeah.
Yeah. Thank you so much for giving me this opportunity. It was quite a nice I was figuring out recently while navigating on LinkedIn regarding cleaning validation is when we are dealing with equipment grouping, as such, I saw some experts commenting out, and I agree to them that equipment grouping is a very contradictory scenario when it comes to contamination contamination issues, cross contamination basically, when having especially multi-product facilities. So equipment grouping is something that needs to be avoided. As such, I got to know, and I wanted to ask Thomas that what is his take on equipment grouping and how safe and how dangerous it can be. Yeah, thank you.
Thank you for that question. Because if you look to this equipment grouping, it is always outlined that they should be as similar as possible, and then you have these kind of scenarios where you are getting people saying, they are quite similar, and I will say that the biggest of like IBCs, intermediate bulk containers, they have 500 liter, they have 1,000 liters, and then they say, oh, the 1,000 liter one is much more harder to clean because the dimensions are wider. I consider this as worst case. But if you look really to the details, then it is something where you might now say, is it really the same? And the worst experience I had lately was I was on a site, they had different mills. That was even also an API production. They had mills on site, 5 different ones, and they considered them all to be the same because they are all mills. But then we said, but you have really not the same even manufacturer. They are in the construction a little bit different. But we clean them manually in any way. So this is why we want to consider them all in one group as mills. And then you said, I would say, you can do that only if you say we have all the same cleaning process with it. And we will make then the identification, for example, with the kind of how do you call it, with the kind of limits what you set for the mills, and then you have the same cleaning process and you have the sampling, and then you can identify, okay, for the same cleaning process using different equipments, this is the worst case, and you don't need to validate everything. But if this is manual, you can even not really validate that even in a very good way. So you must be really careful of what did you do in the past? Who was considering that all the equipments are the same in one group? And what was the rationale behind? Is this still true today? Or can you maybe have arguments that no, the dimensions are completely different, and this is why I will not consider that as one group of cleaning in the future anymore.
Yeah. And then there is another question, Thomas, which is around animal health products. So we know that there are now lots and lots of facilities manufacturing animal health products. So the question is that how do we determine PDE value for products used for multiple species of, let's say, dogs and cats, which has different maximum daily dose, which I think can vary with because of the species and age. And I think this could also be applicable for even the human drug products, where the maximum daily dose can vary. So how do we take that into account for determining the PD / AD value for particularly for the animal health products?
So we usually recommend to make the calculation for all the different circumstances and see what is the worst case, because that is what you would need to make sure that you are covering the worst case with your limit setting. So you take I would say, the lightest animal with the highest daily dose. That would be considered as worst case, but you will see it when you calculate your safe surface limit. And this is what can tell you. But you should have like a metrics where you say, all these animals can get it. These are the assumptions of the weights of the body of each of the species. And this is the resulting limit. And then you need to select the worst case for your validation studies, because that is, you cannot determine who will get this product. If there is a chance to say, oh, in this package, you will only get it to the horses, and in this package, it is only going to the cats, well, then it's fine. But for cleaning validation, when you make your product then you have made to make the paste, and you do not know where in what package it ends up. So this is why you need to make the worst case for cleaning validation and then the limit setting based on that.
Thank you all of you for joining the webinar today, and almost all of you actually stayed back till the very end, which is always a really good sign. Everybody likes what we have to say, Thomas, on this topic.
Thank you.
So thank you so much for bringing all of this essential knowledge and insight to this webinar today. So with that, we'll close the session. Thank you all.
Thank
Have a
you
good
for
day.
organizing, and have a good day. Bye-bye.
Bye.